A very good afternoon. So today we'll be discussing on a very commonly used technique in the research labs and in the clinical setting, flow cytometry. So the first talk will be given by Dr. Shashidhar. Will be discussing on the basics of flow cytometry and its use in research. And then that will be followed by Dr. Bala Sahib from Hematology, who will be speaking on the clinical applications. So,
Yeah, thank you, Dr. Ru. Welcome to Clinician Scientist Interactive Session. Today I'm going to talk about basics and clinical research of cytometry, which we use regularly in our research activities. So before going into the basics and applications, research applications, so in any biological material, so we want to analyze any 2 to 3 parameters or something like that. So we can use the basic technique like ILC, that is immunohistochemistry. So where we stain the sample using different fluorochromes and will visualize under the microscope. But we want to use more than 4 fluorochromes or 4 different parameters we want to study. The best technology we have is flow cytometry. So the flow cytometry, so the basic information which we want to get from the flow cytometry instrument is like whether the cells of interest are positive for a given marker. But the basic challenge in this flow cytometry is revealing the dim events or rare events which is difficult to identify in the flow cytometry. So where the technology has developed, so we can easily identify the rare population and also current population within flow cytometry. So population which is in abundant or more numbers, you can easily available, visualize using the programs, for example, like CD4 or CD8, which are a high number in the sample. So easily identify. Even the less numbers like B cells, we can identify. But the rare population, like T regulatory cells, if you see, 0.001% in the human blood. So it is very difficult to identify T regulatory cells. So we need to have proper fluorochromes and bright fluorochromes to identify this rare population. So why flow cytometry? So flow cytometry, which is an extremely powerful and versatile technology for most of the biological, because it is very fast. You can more than tens and thousands of particles events second, and the large amount of data can be generated, which allows us to do robust statistical analysis, and also a lot of information also will be gained, and many physical and fluorescent parameters are measured simultaneously, which allows us for the resolution of distinct populations and cellular function complex and heterogeneous mixtures. So this data, which is generated by flow cytometry, is qualitative, quantitative, and unbiased. So using this flow cytometry, we'll get pure population, can isolate or sort pure population of sterile live cells of interest, which can be retrieved for downstream applications such as culturing or functional analysis, transplant or imaging, and omics using DNA and RNA sequencing, proteomics, and metabolomics. So basically, the flow cytometry, it is a laser-based technology used to analyze and measure physical and chemical characteristics of cells or particles in fluid mixture. It provides rapid and quantitative information about cell size, complexity, and fluorescence intensity, enabling us various applications in research and clinical settings. So what is flow cytometry? Flow cytometry, flow, it cells in motion, cells, cyto is cell, and metry is measure. So measuring the properties of cells while in a fluid stream. So flow cytometry basically it consists of 4 systems like excitation optical system, fluid system, emission optical system, and electronic system. So we have to have a laser source where this laser will excite the cell, where the cells will emit the light, which will be captured by the PMT detectors. These PMT detectors will analyze and transfer the data into the uh, computer. So these digital analogs will be analyzed using the softwares, different softwares actually. So in flow cytometry, what are the particles we can analyze? I mean, like range from 100 nanometers to 100 micrometers, we can analyze flow cytometry using the flow cytometry, like various types of cells or organelles and also red blood cells and all. We can analyze using the flow cytometry. So what is the basic function of flow cytometry? Basic function is like when we give the input of mixture of unlabeled and labeled cells, output is labeled cells. Percentage number of labeled cells we can get using the flow cytometry. And the basic mechanism is like any biological sample, we stain with or label with a fluorescent marker, and cells move in a linear stream through a focused light source, that is laser beam, and these fluorescent molecules get activated and emits light that is filtered and detected by sensitive light detectors, usually photomultiplier PMT tubes. And these signals will be converted to analog fluorescent signals into digital signals. So we have different applications in clinical setting and research setting, like we can in biomarker discovery or drug discovery, or even in cellular studies and so on. So that will be discussing later in the coming slides. But coming to the advantages and disadvantages of the flow cytometry, the major advantage of using the flow cytometry is like we can analyze the samples in high speed, like more than 1 lakh events per second. Also, we can analyze it depending on the flow rate, and also it allows us to deal with information which we are collecting using the flow cytometry, and we can do the simultaneous analysis with multiple parameters with the help of computerization in a large scale population of which provides the statistically significant information, and also it identifies the very rare population, and it will also allows us to study the cell by cell fluorescence study, and even we can sort the cells in large quantities, like 17,000 cells can be sorted per second using the flow cytometry, and is portable equipment. The disadvantage of using the flow cytometry, yes, by using the single cells, so cells are used, so we may lose the architecture of the cells, and also these results obtained from the flow cytometry are depending on the specimen collection and also process, how we are processing the sample. And the quality of the cell labeling is affected by viability of the cells. When we use the heterogeneous population, which contains fragile subpopulations, which might be damaged during the process, leading to false interpretation. And this flow cytometry is very expensive, and it is a sophisticated instrument also, but it requires management of highly trained specialists and also ongoing maintenance by service engineers, and it's a complex instrument. It is prone to a problem with microfluidic systems, and also requires warm up, and maintenance is very high using for this flow cytometry. Definition of flow cytometry. The discovery of microscope in 1590 by Leeuwenhoek till 1972, where the Heisenberg has discovered the flow cytometry for the first time. So in 1960, the Kaminsky at IBM has developed a spectrophotometer, rapid cell spectrophotometer, which was used to measure the nucleic acid content and cell size using the arc lamp source, and the data was analyzed using the computer. So with the IBM support, the Heisenberg has developed a prototype called FACS, that is fluorescence activated cell sorter. After 1972, this technology has been overtaken by the industry, like BD, Beckman, and Arthur, and they have developed various flow cytometry equipment for different applications actually. So the different types of flow cytometers we have, like traditional flow cytometers, cell sorters, acoustic flow cytometer, spectral analyzers, imaging cytometers, and mass cytometers. So the traditional flow cytometer, which uses basic optic, fluidic, and detective systems where we can analyze basic, cell functions and phenotype characteristics actually. But by using cell sorter based on the interest of type of interest cell, we can sort the cells using the flow cytometer. So the difference between conventional and spectral flow cytometry. So the important difference between these two are like the dispersion. Where the conventional flow cytometer and spectral flow cytometer. In conventional flow cytometry, the optic blocks and reflect and transmit a photon based on its wavelength, whereas in spectral flow cytometry, the dispersed photons according to the wavelength. The most common types of dispersive optics are prism and gratings. Why we are using this spectral flow cytometry means for studying the 2 to 3 parameters, we can use conventional flow cytometry. But to analyze major or multi-parameter analysis using different more than 6 or 10 fluorochromes, so we have to use spectral flow cytometry. So along with, in this acoustic flow cytometry, the basic problem with the flow cytometry is the clogging of the cells. The sample will be clogged, where the cells will be aggregated, and the flow cytometry will be blocked. The nozzle will be blocked. So to overcome this problem, they have developed using the ultrasonic waves were introduced into the flow cytometry chamber, where the cells will be passed through the flow cytometer nozzle without any clogging. So this is the advantage of using the acoustic flow cytometer. And also we can analyze high input, we can use high sample number also can be analyzed using the acoustic flow cytometry. So this is the upcoming technology where along with fluorescence microscopy and flow cytometry, both combining together, they have developed the imaging flow cytometry, which allows us to rapid analysis of sample for morphology and multi-parameter fluorescence in both single cell and population level. Then mass cytometer, combining the time of flight mass spectrometry with flow cytometry, we can analyze the samples. Here we are not going to use fluorescence and fluorochromes. Here heavy metal ions are tagged with the antibodies, and these metal ion tagged antibodies will be labeled to the cells, and that will be analyzed in the mass cytometer. And today we have advanced flow cytometry where they use detectors like regular detectors or PMT detectors. We have also avalanche photodiodes and silicon photodiodes also using for the detection. There will have stable emission factor. So this is the latest flow cytometer available in the market, like where we can analyze more than 60 parameters using 9 lasers actually. So here we can use up to 55 fluorescence markers and 5 scattered parameters can be analyzed using this Bigfoot spectral cell sorter, recently launched by Thermo Fisher. And we have in the market, we have more than 10 number of flow cytometers are available, where we can analyze using different fluorochrome parameters we can analyze. Coming to fluorochromes are important in the flow cytometry. When fluorescence is described, it is in terms of excitation and emission. Fluorochrome is excited by the laser, a defined excitation wavelength, at which time the molecule absorbs light and photons. So then it returns to the ground state where the fluorochromes emits the photons, less energy and longer wavelength. The difference in the two excitation emission wavelength is called Stokes shift. The fluorochromes exhibiting larger Stokes shifts are generally more desirable for fluorescent research, as the emitted light can be easily distinguished from the exciting light source. So these fluorochromes can attach to the antibodies or proteins or free molecules that becomes fluorescent when bound to the target or biological conditions. So this phenomenon is called Jablonski phenomenon. So we have different types of fluorochromes available. Actually, low molecular weight compounds like porcine, rhodamine, or coumarin, which are low molecular weight. And also high molecular weight compounds like picoerythrin and phycocyanin. These high molecular weight compounds are isolated from the bacteria and other algal sources. And coming to tandem dyes, these tandem dyes are combination of low molecular weight and high molecular weight compounds which are covalently bound, where tandem dyes are prepared. Like for example, like PE picoerythrin Cy7, which is a tandem dye of picoerythrin using picoerythrin and cyanin, so which will have the brighter emission, where we can detect the rare population also using tandem dyes. And the other one is like quantum dots. These are 2 to 10 micrometers nanoparticles actually, which are attached to the fluorescent molecules. The other one, fluorescent proteins, the natural available fluorescent proteins like GFP, YFP, and fluorescent proteins and fluorescent proteins, they're naturally available. So this will help us in transfection studies, where we can transfect the cells for transfection studies. And polymer dyes, which are now upcoming fluorescent markers available. So these polymers, with depending on the length of the polymer, will get the excitation wavelength, where the stable wavelength can be analyzed using the polymer dyes for multi-parameter analysis. And even metal conjugates, like for mass spectroscopy, where the metal ions like these are lanthanoid derivatives, where we use for metal conjugates, and nucleic acid dyes for happy and all we use for nucleic acid studies, and proliferation studies like CFSC, carboxyl fluorescence, for proliferation of the cells, and viability dyes like host and PI, which we use for the cell studies actually. So in evolution of fluorochromes, right 70 organic dyes were developed, then phycobiliproteins, tandem dyes, two dots, then polymers were developed. We have more than 367 CD markers are available, which will use for intracellular analysis and all. So for conducting a cytometry experiment, so we have to consider 3 parameters actually. That is pre-acquisition, acquisition, and post-acquisition. In pre-acquisition, we have to look for the cell viability, cell preparation, antibody panel, fluorochrome selection, staining procedure, and control. And acquisition, like instrument configuration, maintenance, and proper controls, with quality control and quality analysis. And post acquisition, gating template and data reporting with proper gating controls. So what are the parameters we have to consider before conducting the flow cytometry experiment? So basically, what is the cell type we are going to study? What are the antigen characteristics? Based on this, we have to choose the right fluorochromes which are compatible with the instrument which we are going to analyze. So all are interlinked. In the experimental controls, we have to use proper experimental controls which will help us to resolve issue in staining, like isotope controls which will help in identifying the staining issues, unstained controls which highlight the background or autofluorescence of the system, single stain controls which will allow you to perform the quality control of the compensation and assess the resolution impact, and fluorescence -1 FMO controls help in identifying the gating boundaries and illustrate the potential impact over the spill. So this is the example where the spillovers we are going to compensate using the FMO. So here the PE, phycoerythrin, stain to the CD4, we want to stain with the CD4, but whereas PE Cy7, PE Cy5 will spill over into the PE channel actually. So to overcome that, first we stain the cells, except with the PE channel. So we use this FMO gate. Later, the cells will be gated for CD4 PE. So like this, we can get the exact population using the isotope controls. So for selection of fluorochromes, what type of fluorochromes we have to select for our experiment? So we have to design a panel which will give us the proper interpretation, and critical populations of interest can be obtained using the fluorochromes. The main goal is to minimize loss of resolution due to spread from the fluorescent spillover of co-expressed antigens. So how to avoid this spectral spillover? When antigens are co-expressed on the cell, avoid significant spillover of bright marker into the dim marker and spread the antigens across the many lasers as possible. And fluorochromes that are excited more than one laser, high spillover, like for example, like AMSAN excited by the violet and blue laser spills into the FITC detector, and P-Sci-Fi excited by blue and red laser spills into the APC detector. And we have to consider for tandem dyes also for donor emissions. So how to minimize this spillover into the populations of interest? Suppose we have the population of where the markers we are using with different populations where different markers are expressed, like B cells or T cells. They have different phenotype markers, so we can use any fluorochrome markers and we can easily detect the CD19, that B cells and CD4 cells or CD3 cells. But whereas cells which are co-expressed, like CD3, the basic lymphocyte marker, on CD3 we detect CD4, CD8 markers. So for co-expression markers, so we have to carefully choose the fluorochromes to analyze the data actually. So the markers are co-expressed. So we have to see the relative expression. Either, for example, like here, I illustrated with A and B. So the A is overexpressed with B. So we have to consider B as a bright, fluorochrome because B expression is very low. So we have to choose for expression of B marker, we have to choose the bright fluorochrome. And for A, we have to use dim fluorochrome. And vice versa with B and A. So when we choose the reagents properly, so we'll get the proper expression of different markers. So coming to compensation, so most of the fluorochromes which are excited by laser having long emission curves. So a flow cytometer will have filters which optimize for the specific wavelengths of light. So, but unfortunately, overlapping emission wavelengths from one fluorochrome will spill over into the filter of another. So this spillover can be corrected mathematically by process of compensation. So the compensation is not dependent on brightness, it is dependent on the emission of spectra of a fluorochrome. So once the compensation is set, the compensation values remain applicable for successive dim or bright samples which subtracts percentage of fluorescence intensity. So for example, you can see the different lasers have different fluorochromes. UV laser, so we can use either DP or host 3778 fluorochromes where they are distally have emission wavelength. And even in blue laser, so we have FITC and per CP Sci5.5 where they are distally distal wavelength emission wavelength spectra. So here we can use in laser we can use either FITC or per CP. But in violet laser, you see there is a overlap of or spillover of the fluorochromes. So here we have to use the compensation controls where the spillover should not overlap with each emission spectra actually. So this is one of the examples where compensated and compensated. Non-compensated, you see there is a mix of viability dye with CD3 dye. So here we are getting for CD3 and viability using. It's a mixture of, both are mixed, populations are mixed. But when you do the compensation, you can separate the dead cells with high CD3 population. Even if you analyze the cytokine, one of the examples, CD8 cells, if it are not compensated, so we cannot distinguish the problem positive from a positive cells with CD8 cells. So here you can see different gamma cells properly using this compensation. So here we do, the difference between uncompensation. So the basic thumb rule in compensation is whatever the populations we are getting in the flow cytometry, populations has to be on straight line. The center of the population should match with the other population. So this is called correctly uncompensated. The population will be in different compensated or undercompensated or overcompensated. So panel development. So for example, for study of deregulated cells, so we can study basic parameters like memory status of the T regulatory cells or phenotypical characteristics of different parameters we can use. So you can differentiate them for central memory, effector memory, and effectors of using these markers. How to choose the proper fluorochrome marker for this fluorochrome for these markers. For example, like in this experiment, for Treg phenotype, I have chosen different parameters. Out here. So the red color mark highlighted parameters, they are bright fluorochromes actually. You see BE, BV711, BV421, the bright fluorochromes. Where the rare populations like CD25, FOXP3 or CD127 in T-regulatory cells, you can easily identify in T-regulatory. Also the origin of T-reg, either thymus origin or preferred origin of the T-regulatory cells, we can easily identify using CD31 and CD39. In bright. So this we have to carefully, separate the fluorochromes for. We have to design the fluorochrome panel for this experiment. So the data visualization, we can visualize the data using different platforms. So here we can use either using dot plot, color, and different display visualization we can do. Important strategies like gating strategy. How we are going to separate the cells after labeling the cells. Acquire the sample. So without gating, sequential gating or progressive gating, cannot distinguish the type of cells in the top, top row here. Here we cannot distinguish which type of cell it is because it's a mixture of all cells. But when we do the sequential or progressive gating, so we do the live cell gating and later singlet like single cell gating, and later we'll go for either CD11B. Specific type of cells we can easily identify using the gating. So here is a spectral flow cytometry for high dimensional analysis for using more than 40, 50 fluorochrome parameters. So we have to use a high dimensional analysis. So we have different fluorochromes are available with different lasers. Here, high lasers that you are having. So more than 60 fluorochromes are available in this panel. This is one of the example where we use, where they have a color panel for basic phenotype markers in purple red mononuclear cells. So using UV, green and red lasers. This is the data with single staining. We can get data for different cell types like connective T cells, monocyte, T cell, gamma delta cells, related T cells, and so on. Different types of cells can be analyzed with single stain of single panel actually. So coming to flow cytometry applications, you can use it for academic and clinical disciplines. You can use for cell counting, characterization, analysis of cell markers, for phenotype markers. We can use for cell sorting, multi-parameter analysis, detection of aberrant cells or abnormal cells, investigation of immune cell function, cell signaling analysis, pathological illness, diagnosis and so immunophenotyping, transfection efficiency, measurement, cell cycle analysis, fluorescence intensity, cell proliferation, membrane potential, live rate bacteria. So next I will couple of the clinical applications using flow cytometry here. Analysis of leukemia and lymphoma cancer. In detail, Dr. Bala will explain presentation. So for immunophenotyping of flow cytometry is an important tool for the diagnosis and staging of the patients with hematological disease. As much as we can different markers to distinguish the different cancer markers actually, and even we can detect use this flow cytometry of minimal residual disease (MRD) is defined as a disease beyond the limit of morphological detection using conventional microscope. This is one of the for diagnosis. In the diagnosis after treatment, take the very rare populations of MRT positive and MRT negative, we can easily distinguish in the flow cytometry. And for immunophenotyping of lymphoma and lymphoid leukemia conditions using different parameter sequential markers we have to choose, like CD5 negative or CD19, CD, I mean, different parameters we follow and to identify the types of cancers, leukemias and lymphoid leukemias. And this is other phenotype like ALL and AML using different markers. We have to choose the right panel for distinguishing between, I mean, the difference between ALL, and this is the gating strategy where essentially gate for the CD13, CD30, 71. And the other cytometry is stem cell enumeration. So for once we mobilize the stem cells from the bone marrow using GCSF, we have to identify the proper enumeration of stem cells using the cytometry, and we can, and also in solid organ transplant, we can check for pre and post operative for where we use the recipient serum and donor lymphocytes, we can detect the antibodies there prior to organ transplant. So where we'll get to positive and positive, can distinguish easily, and we can use this flow cytometry for regenerative therapy for different stem cell therapies we use for conditions, and also fetal maternal hemorrhage also there. Anti-fetal hemoglobin distinguish fetal examples. Also, we can use cytokine profile monitoring, promise for sepsis evaluation, and also and microbial infections where we can identify easily anti-inflammatory cytokines using the this cytometric bead assay, and we can use clinical conditions like infection, immunodeficiency diseases, paroxysmal nocturnal hemoglobinuria, and reticulocyte analysis. And this flow cytometry also used basically, which enables us for the multiplex to phenotypic primary screening of cells, screening of sources for every, biological drugs like antibodies. For example, like flow cytometry of critical importance in the CAR T cell therapy development, which includes patient diagnosis, characterization of the manufactured product, and infections, diseases. Also, we can identify for
Good afternoon, everyone. After listening to Shashidhar sir's presentation, my topic is very small, and I'm just covering clinical applications of the flow cytometry and how we what samples to take when ordering the flow cytometry, and in what illnesses we use flow cytometry majorly to diagnose hematolymphoid malignancy and other indications. I will be covering samples used, diagnostic uses in benign disorder, malignant disorder, use of flow cytometry in the risk stratification, advantages of the flow cytometry, and drawbacks. Basically, Shashidhar sir has covered a flow cytometry in detail, the principle of flow cytometry. Basically, in simple language, flow cytometry is just analysis of the cells with the help of markers located on the cell surface when while they are flowing through the cells flow cytometer or cell analyzer. The basic the basic model has been explained by Shashidhar sir. What samples we use when we order the flow cytometry test for a diagnostic purpose. The commonly used sample is a bone marrow aspirate for all the hematolymphoid malignancies. Peripheral bloods also can be used for a diagnostic purpose. When we want to diagnose acute leukemia and peripheral blood, if it shows a high WBC count with lymphocytosis or abnormal cell, even the peripheral blood sample also can be used. Cerebrospinal fluid also can be used for a diagnosis of hematolymphoid malignancies. Provided cerebrospinal fluid has adequate cells, it can be also another reliable sample for diagnostic purposes. Ascitic fluid and pleural fluid also are used in flow cytometer, and cell suspension created from the fine needle aspiration cytology also can be used for running a flow cytometry test. Basically, in clinical practice, we have a dictum that if we have a tissue, if we have a tumor mass, we should biopsy it. But if tumor mass is not accessible and fluid is accessible, like mediastinal mass is there, and along with that we have a big pleural effusion, then we can tap a effusion instead of biopsying and send for a flow cytometry. That is the easiest way. And then blood. If we have a possibility of diagnosis on the blood, we can use the blood for running a flow cytometric test. Clinical uses of the flow cytometry. Diagnostic uses. In malignancy, flow cytometry is a mandatory test for diagnosing acute leukemias. Flow cytometry also can be of diagnostic use in the lymphomas, particularly low-grade lymphomas. It doesn't have that much value in high-grade lymphomas because high-grade lymphomas, what markers pathologists use are mostly they are available on immunohistochemistry. Acute leukemias, flow cytometry is commonly used in to diagnose lymphoid leukemias and myeloid leukemias. Lymphoid leukemias, both acute and chronic, can be diagnosed on flow cytometer. Myeloid leukemias, it is mainly used to diagnose acute leukemias. Chronic myeloid leukemia doesn't need a flow cytometry for a diagnosis. We just use a different method to diagnose chronic myeloid leukemias. Coming to acute leukemia, we have three different subcategory of acute leukemia grossly. One is T cell type, then B cell type, and then mixed phenotypic type. So there are classical markers which are used to diagnose each and every leukemia. Like say, if we have a B cell leukemia, we mainly look for CD10, CD19, and along with that CD20 and other markers. When we are suspecting T cell leukemia, pathologists will look whether T cell markers like CD3, CD5, CD7 are expressed. When we have suspected myeloid leukemia, that time they use different type of markers. Supposing we have a mixed phenotypic leukemia, there are specific guidelines like European Leukemia Net guideline where a particular marker is used if it is positive. Like say we have a B cell phenotype mixed with a T cell phenotype. In that we should have there is a scoring system like CD19 positivity or CD10 positivity or TDT and CD3 positivity. For if there is a myeloid, then MPO and CD33 are used as myeloid markers. So this particular flow diagram shows that it is a B cell leukemia, with aberrant expression of CD7. As sir has explained that all the flow cytometer can the various colors can be used to interpret the different cells. Here the cell population which is falling above the line or in the extreme right quadrant is considered as positive. This particular flow cytometer is a classical diagnostic of chronic lymphoid leukemia, where cells are positive for CD20, CD23, CD5, and the positivity is shown by the shown by green signal. Coming to acute myeloid leukemias, flow cytometry wise, all the leukemias have almost same cell expression except AML-M3. AML-M3 can have a certain characteristic of flow cytometry. AML-M3 CD33 will be strongly positive along with MPO, which is strongly positive, and most of the times HLA-DR and CD34 tends to be dim positive, or they can be negative also. Most of the other subtypes of acute myeloid leukemia, they have almost same characteristic with subtle differences. That details of I am not covering, and it will be confusing also. But we can have clues of different leukemias, and this particular flow cytometry needs to be interpreted along with the genetic analysis. Say we order a cytogenetic analysis, basic karyotype, fluorescent density hybridization, and various nowadays various PCRs are used, plus clinical exome sequencing is also available, which we call as next gene sequencing in clinical situation. Flow cytometry plays a very huge role in diagnosis of low-grade lymphoma, and there is a very nice algorithm developed for how to diagnose low-grade lymphomas on flow cytometry. Here, once we most of the times low-grade lymphomas, they involve the bone marrow and spleen. Most like commonly diagnosed low-grade lymphomas, CLL, mantle cell lymphoma, or marginal zone lymphomas, splenic marginal zone lymphomas. This particular algorithm is very commonly used in the lab to distinguish different type of low-grade lymphomas. Flow cytometry plays a huge role in diagnosis of benign disorder, immunodeficiency diseases, platelet function disorder, paroxysmal nocturnal hemoglobinuria, and autoimmune lymphoproliferative disorders. Immunodeficiency in immunodeficiency disorder, flow cytometry has a major role to diagnose different types of severe combined immunodeficiencies based on which combination of lymphocyte subsets are absent or reduced in number. Along with that, we can diagnose chronic granulomatous disease, leukocyte adhesion diseases, and usually the use of flow cytometry, most of the times flow cytometry use, it's a diagnostic test in the severe combined immunodeficiency, but other diseases it is interpreted in context of clinical presentation and other tests. Nowadays, next gene sequencing is used commonly along with the clinical presentation and flow cytometry to diagnose primary immunodeficiencies. This is an algorithm used by laboratory to distinguish between different types of severe combined immunodeficiency. They are majorly divided into two. There are three subtypes of severe combined immunodeficiency. One is T negative, NK positive, and B positive. Another is T positive, NK negative, and B positive, and B negative with other. Here we order lymphocyte subsets. It can be done on the peripheral blood, and peripheral blood lymphocyte count also should be interpreted along with the flow cytometry report. Other disorders which can be diagnosed by flow cytometry is platelet function disorder. In Bernard-Schauler syndrome, flow cytometry plays a diagnostic role. When we have a patient with a bleeding phenotype, say young female where attending menarche presenting with the menorrhagia, and if she is found to have giant platelets on the peripheral blood smear, at that time we we suspect that Bernard-Schullery syndrome is can be possibility, and we can order a flow cytometry to find out the antigen which can be defective on the surface of the red cells. Flow cytometry is also used to diagnose paroxysmal nocturnal hemoglobinuria. Most of the times, the fresh sample is required, and it is very reliable to diagnose platelet function disorders by flow cytometry. It has to be drawn from the patient and it should be analyzed on the same day. Same is applicable for paroxysmal nocturnal hemoglobinuria also. The fresh sample gives us a correct report, because as the longer the sample stands, the antigen on the red cells can be get deteriorated, or flow cytometry will not be able to pick them up. Flow cytometry nowadays is playing a huge role in monitoring the response to the therapy. While we treat various type of hematolymphoid malignancies, particularly acute lymphoid leukemia and acute myeloid leukemia, we call it as a minimal residual disease. Nowadays, the terminology is measurable residual disease. This particular test also is done using previously eight color flow cytometer was being used. Nowadays, people are using 10 color flow cytometers, and it is very well established monitoring tool in case of acute lymphoid leukemias. We do the MRD assessment at the end of first induction chemotherapy, and if it is less than 0.1%, then it is called as very good response. Flow cytometer can diagnose one abnormal cell in 10 lakh cells, so the sensitivity is very high. Previously, when I started as a student in hematology, we used to use only bone marrow smear as a marker of controlling the disease. But gradually this MRD has come into the picture, and there are various algorithms developed to treat lymphoid leukemias. Then the MRD is assessed usually at the end of induction, that is called as day 37 MRD. Then second MRD is assessed at day 52, that is called as second MRD. If second MRD is positive more than 0.1, then patient has most of the time he has a poor prognosis and needs a stem cell transplant. MRD is also getting established in monitoring the disease response for acute myeloid leukemia. Many centers and the central referral laboratories are offering the MRD for acute myeloid leukemia. Our center also is going to start monitoring MRD gradually for acute lymphoid leukemia and myeloid leukemia. MRD's role has been also noted recently in chronic lymphoid leukemias. In my clinical practice from 2019, after I passed out from I postgraduated in hematology, CLL as of now I have not used personally, but there is a lot of literature that MRD can be used to monitor the chronic lymphoid leukemias. It also can be used to monitor hairy cell leukemia, mantle cell lymphoma, leukemic phase, mantle cell lymphoma, leukemic phase. It can be also used in splenic marginal zone lymphoma. There is only one lab in Mumbai who does all the MRDs in lymphomas. Many central laboratories are offering gradually disease monitoring by flow cytometry. Flow cytometry has a big role in monitoring the immunosuppressions. Not only hematology uses flow cytometry. There are other branches who also can use flow cytometry, like rheumatology or neurology, when they treat various autoimmune disorders and they want to see whether the immunosuppression is working. Particularly after giving rituximab, if they want to see whether rituximab is really working or not. So they order something called as lymphocyte subset analysis, and they see whether rituximab has suppressed adequate number of B cells, and there is adequate suppression of the B cells. Flow cytometry nowadays also can be used to monitor whether patient's immunity has recovered after the stem cell transplant, particularly allogenic stem cell transplant. And there are various time points when we should do the flow cytometry to monitor the immune reconstitution. Uh, uh, liter- uh, Theoretically, they are recommending flow cytometry to be done to monitor immune reconstitution at the end of 3 months, 6 months, and at the end of 1 year, to see whether NK cell function has come up based on the NK cell markers by flow cytometer. As Shashidhar sir has discussed, flow cytometry also can be used to enumerate CD34 before the stem cell transplant, and that is a common. This is we are doing in house. We have a test available here, and this CD34 estimation also is being used by neurosurgery people to do their clinical research in stem cell transplant in cord compression or the cord disorders, spinal cord disorders. Coming back to advantages, it is very sensitive and very specific for diagnosis of acute leukemias, low-grade lymphomas, and it is very sensitive and specific marker of disease measurement. Nowadays, MRD is possible by monitoring the molecular markers also and the cytogenetics markers also. Suppose if I have a patient of acute lymphoid leukemia, and I have done and he is say translocation 922 positive. MRD can be monitored by flow cytometry also, and it can be monitored by molecular techniques also. Flow cytometry molecular techniques are far more sensitive and far more accurate in monitoring MRD than flow cytometry, but flow cytometry still stands a major ground. So advantage is sensitivity and specificity is very high. Turnaround time is very less. Suppose if we collect a sample, give the sample to the lab, say practically before 2 p.m. or 3 p.m., then lab can process it on the same day and give the report by say evening 5-6 p.m. So turnaround time is very less to diagnose hematolymphoid malignancies. Disadvantage is this: it's a costly test, but it's a must test. All the acute leukemias should have a flow cytometry done, should be done. It needs a expert who runs the machine, like technical support, and it needs a trained person for analysis. Otherwise, most of the times clinicians will not be able to interpret the details of the CD34 enumeration and flow cytometry. That needs to go through the experts for analysis. So that's why most of the centers don't have their own flow cytometer. We have a flow cytometer being used in research, and now pathology also is offering the test. With this, I conclude my presentation. Thank you very much.
These speakers are covering the spectrum physics. Is there any question? Back talk, sir.
Mass is not accessible, and we have a high suspicion of lymphoma. Ascitic fluid also gives a diagnosis, or pleural fluid also gives a diagnosis. For example, recently one case got admitted in pulmonary medicine with huge mediastinal mass, and the patient was having severe respiratory distress. We did a pleural fluid flow cytometry. It turned out to be a follicular lymphoma. Follicular lymphoma, the report came within 26 hours. So most of the times, low-grade lymphomas and even when it is infiltrated, uh, the pleural fluid or ascites or effusion, if it is caused by malignancy, it can be diagnosed through the pleural fluid.
We do flow for all the cases where there's a strong surgical in the pleural and ascitic.
When hematolymphoid malignancy is highly suspected, then we should do. Couple of the tests before we started our flow cytometry here in we have sent to Apollo for pleural analysis. This particular case of follicular lymphoma was diagnosed in Apollo. Before that, we used to send to OncoQuest. CSF also flow cytometry can be used, but then when there is a dilemma in diagnosis, say there are five lymphoid cells, more than 5 cells, but they are looking normal or suspicious, then we can run a flow cytometry. If there are less than 5 cells, then we usually don't recommend on the CSF.
I was just thinking maybe we should validate fluids with positive lymph node, pleural or ascitic fluid. If, during your rounds, if you can give us, they will be as for us to validate pleural and ascitic fluids for diagnostics.
We can do that actually. If we have if there is a difficult sample and small node is there, even we can try FNAC and we can try through that, but it's not ideal.
No, in well-formed cases with ascites, if you can give some fluid for validation in the coming months, improve the, because one odd case means we also won't be sure if you're dealing with the correct thing.
And next Thursday we'll continue the AI session. I think. Thank you.